Good morning. This video will contain a rapid review of the main concepts that you have to know for your semester 1 test tomorrow, inshallah. I'll go slide by slide and tell you as much as I can what kind of questions you might encounter on the test that are related to the content of the slide. Of course, for this slide, you are supposed to know the labeling of the urinary system. So one of the important questions is that you might have a drawing on the test, and that drawing might be unlabeled. What you have to take care of is that the kidney on that side is the right kidney, because as we said before, you consider that you are looking at a person in front of you. So that kidney that is to your left or towards your left hand will be the right kidney, whereas the other kidney will be the left kidney. So right kidney, left kidney, right ureter, left ureter, urinary bladder, urethra, and sometimes we have a labeling for a thick muscle ring here that we call the urethral sphincter. Okay. Of course, you might have to, or you might be required to label the major vessels that are connected to the kidney. So how would we know that this is the aorta? We would know it from the direction of blood flow. So if the arrow is pointing downwards, then the blood is going downwards into the large artery and towards the kidney. And if the arrow is pointing upwards, then this is the inferior vena cava, and blood is moving upwards towards the heart, because the inferior vena cava returns deoxygenated blood to the heart. And then I might ask you another question about the direct blood supply of the kidney. So the direct blood supply of the kidney will include the renal artery. So this is the renal artery, and the other one is the renal vein. Again, since you don't receive a color printed exam sheet, then you wouldn't have a difference in color. You will just know from the direction of the arrows. If you don't have arrows and you have to label the diagram, then you would say renal artery and renal vein together. Now you might also be asked about the difference between the renal artery and the renal vein. Of course, if we draw the kidney like this, so the renal artery will carry blood to the kidney, whereas the renal vein will carry blood away from the kidney. The renal artery will contain oxygenated blood, whereas the renal vein will contain deoxygenated blood. These are the two major differences, and you might, of course, be required to explain what oxygenated blood is and what deoxygenated blood is. So oxygenated blood is blood that carries more oxygen and less carbon dioxide, and deoxygenated blood carries less oxygen and more carbon dioxide than oxygenated blood. But in addition to these two major differences, you could say that the third point is in the differences is to talk about the nitrogenous waste substances. Of course, the renal artery has more nitrogenous waste than the renal vein, and I might ask you why. Because the renal artery carries the blood that has to be, or that is going to be, filtered in the kidney, and the renal vein carries back or away from the kidney the blood that has already been filtered from a good deal of the nitrogenous wastes. And then we could be asked, or we could be required to answer a question about the need for a urinary system. Of course, the need for a urinary system is similar to the need for any other excretory system in the body, and it is to get rid of the unwanted waste products of metabolism and to keep the required substances. And the purpose of these two actions is to achieve homeostasis. So the main purpose of the urinary system and all the excretory systems is to achieve homeostasis. How do the excretory systems achieve homeostasis? By two means. Number one, getting rid of the unwanted waste products of metabolism and keeping the required substances within the optimum range. Why do we want homeostasis? Why is homeostasis important? First of all, let's define homeostasis. What is homeostasis? Homeostasis is to keep the internal environment of the cell constant. And why do we need to keep the internal environment of the cell constant? For optimum functioning of the cell. So this is an answer to what is homeostasis, and justify homeostasis is important. But you might also be asked about the aspects of homeostasis, meaning, or examples of homeostasis, meaning that you can be asked about items or values in the body that should be kept constant or within a constant range. So, for example, a normal blood glucose level, a normal water concentration, a salt concentration, acid and base balance, or the pH value of the blood. All these are aspects of homeostasis, okay? You might also be asked about the definition of excretion. I might ask you about this in three ways. I might say define excretion, or what's meant by excretion, or what does the term excretion refer to. So excretion is getting rid of or elimination of the toxic byproducts or side products of metabolism, and actually of catabolism, okay? You might also be asked about the definition of metabolism. What is metabolism? Metabolism includes all the biochemical reactions that occur in the living cells of the human body. You might also be asked about the types of biochemical reactions in the body. So you would say that the biochemical reactions in the body include anabolism and catabolism, and you have to define each even if you are not asked to. So you would say that anabolism or anabolic reactions are building up reactions, and there is no of in between, by the way. Building up reactions. Catabolism includes breakdown reactions. I might ask you about building up reactions. What does building up mean? Building up means that you start by small molecules and you make them react, or you allow them react to give larger molecules. Whereas in catabolism or breakdown reactions, you start by a big complex molecule or several big complex molecules, and you end up by breaking them down into smaller ones. Furthermore, I might ask you about examples of anabolism. So you would say, for example, if we take small amino acid molecules to build up large complex protein molecules, this is one example. Or if we take small monosaccharide molecules like glucose and we build up a complex carbohydrate like glycogen. Or finally, for fats, you might give a third example, which is to take glycerol and fatty acids, allow them to react and form fatty acids. These are three examples that you might give for anabolic reactions. But I might give you a reaction and ask you whether this reaction is anabolic or catabolic. For example, if I give you, amino acid 1 plus amino acid 2 gives rise to a protein, this would be an anabolic reaction. But the reverse is catabolic. So if I give you a reaction where you have an amino acid that is broken down into urea, ammonia, and uric acid, then this would be a catabolic reaction. If I give you a molecule of glucose that is broken down into carbon dioxide, water, and energy is released, this is another catabolic reaction. If I give you a fatty acid molecule that is broken down, a fat molecule that is broken down into fatty acids and glycerol, then this is a catabolic reaction. So whenever you are given a chemical equation and I'm asking you whether this represents anabolism or catabolism, see what the products are, what the reactants are, what the products are, and see which is bigger. If the reactants are bigger than the products, then this is a catabolic reaction. If the reactants are small and the products are big and complex, then this is an anabolic reaction. Here at the end of the slide, we are talking about toxicity of the byproducts of catabolism. So I might ask you about the need for excretion. Why is it important to eliminate the toxic byproducts of metabolism? It's important because if these toxic products of metabolism accumulate in the human body, this would lead to impairment of cell function or a decrease or a deterioration in cell function. Now concerning the functions of the urinary system, as we said, the overall function of the urinary system is to assist in homeostasis, to help all the other organs, to coordinate with all the other organs to achieve homeostasis. But if I ask you about functions of the urinary system in the plural form, thus I mean details, or this means that I need details of the functions of the urinary system, or details of how excretion occurs, how urinary excretion occurs. It occurs through 3 stages. I might ask you directly about the stages of urine formation. So you would say, of course, you all know them. Number 1, filtration, and it should come first, because if you don't filter, you wouldn't reabsorb. So filtration, and then reabsorption, and then secretion. Filtration, you might be asked to define filtration. To filter is to separate. So filtration is the separation of molecules from the plasma of the blood into the cavity of Bowman's capsule through the fenestrations of the capillaries. And you're not supposed to say filtration of toxic substances, because filtration, as we said before several times, it's a mechanical process. It's not a smartly selective process. It just mechanically selects particles, meaning that if the particles are smaller than the size of the pores, they would be filtered, even if they are useful. So it's meaningless to say filtration means separation of toxic substances from the blood. No, filtration is separation of particles from the plasma into Bowman's capsule through the fenestrations. And of course, these particles should be smaller than the size of the fenestrations. But if I ask you about the importance or the significance of filtration to the human body, here you should say that it is important because it allows getting rid of the toxic products of catabolism. And the toxic products of catabolism fall into 3 categories. So you might also be asked about the categories of substances that are excreted or that can be eliminated from the body via filtration. So the three broad titles are products of breakdown of proteins, surplus water and salts, and metabolized drugs and hormones. Now, the products of protein catabolism here, we mean the ultimate products of protein catabolism. I don't mean proteins into amino acids. This is digestion. But when we excrete the very small, when we filter, we don't filter the amino acids. We filter the nitrogenous waste products of catabolism. Like what? Like urea, like uric acid, like ammonia, and like creatinine. Which of these four might have some benefits in the body? Urea, because as you know, urea is not only excreted by the kidney, but also by the skin. So if I ask you a question about the organs that excrete urea, you would say the skin and the kidneys. And in the skin, the presence of urea in sweat is important because it moisturizes the skin. And this can be a justify or a give reason question. Justify, your sweat contains a small amount of urea. This is because when urea is on the surface of the skin, it attracts water molecules. It adsorbs water molecules and prevents their evaporation, and therefore it moisturizes and hydrates the skin. The second substance that, or the second types, or the second category is the surplus salt and water. Why have we put them in one category? Because both of them are essentially useful molecules, but they are filtered because they are smaller than the size of the pores, but we don't lose them completely. We neither lose them 100% nor reabsorb them 100%. We lose in urine, or we eliminate in urine, the surplus water and the surplus salt, and we reabsorb the required amount. And number three, metabolized drugs and hormones. But of course, there is one condition, and I might ask you about this condition. What is the sole condition for metabolized drugs and hormones to be excreted in urine? You would say that they should be soluble in water, because anything that is not soluble in water would precipitate and would cause problems like renal stones and or ureteric stones and inflammation. I might also ask you, where have these drugs been metabolized? These drugs and hormones have been metabolized in the liver. So they are metabolized in the liver, but they are excreted in the kidneys or by the kidneys. I might get you this diagram, and instead of writing the names of the processes, meaning instead of writing reabsorption, filtration, and secretion, I might write process number 1 and process number 2. So I might ask you to name process number 1 and define it, or name process number 2 and define it. Of course, you have to look at the arrows. In process number 1, the arrow passes the molecules from the glomerulus here to Bowman's capsule. You should know that this is Bowman's capsule. Whereas in reabsorption, you return the particles from the PCT here to something that you don't see on the diagram, which is the blood vessel. And I might ask you about reabsorption. So define reabsorption. Reabsorption is moving or transporting the filtered particles that are needed by the body from the lumen of the renal tubule back to the blood. And I might ask you to justify the importance of the process of reabsorption. It is returning the useful substances to the blood in order not to lose them. I might also ask you about, or you might be asked about, these structures in scientific terms. For example, for Bowman's capsule, you might be asked about a cup-shaped structure that receives the filtered substances, or you might be asked about a cup-shaped structure in which, or in the concavity of which the glomerulus lies. You might also be asked in the scientific term about the glomerulus. So it is a spherical loop of blood capillaries that lies in the concavity of Bowman's capsule. And then you might be asked about these two vessels, and you have to look carefully at the direction of the arrows. So the arrow whose head points towards the glomerulus would be the afferent, or we would be referring to the afferent arteriole, and the other one is the efferent arteriole. You might be asked about them in the scientific term, so I might ask you about the small. It would be an easy question if I ask you about the small arteriole, or the small blood vessel that carries blood to the glomerulus. You would say afferent arteriole, and the small blood vessel that carries blood away from the glomerulus, and you would say efferent arteriole. I might also ask you about the differences between the afferent and efferent arteriole. One of them has just been mentioned. The afferent arteriole carries blood towards the glomerulus, whereas the efferent arteriole carries blood away from the glomerulus. But there is another important or very important difference between the afferent and efferent arteriole. What is it? It is that the afferent arteriole is larger in diameter than the efferent arteriole. And then I might ask you about the significance of this particular difference. So you would say that when the afferent arteriole is larger than the efferent arteriole, this ensures high pressure in the glomerulus, high glomerular pressure. And why does this occur? Because you have more input of blood at any time than the output. So again, when the afferent arteriole is larger, this increases the pressure inside the glomerulus. Why? Because the input is more than the output at any time. And how is this useful to the kidney? When glomerular pressure increases, this means that the process of filtration increases. And now comes another question that is related to this diagram, which is, what are the factors that help the process of filtration? So I'm asking here about the factors, the factors that help the process of filtration. Number one, the high glomerular pressure. We have just mentioned it because it helps squeezing out the particles. And number two is the presence of the fenestrations in the capillaries, because if there were no fenestrations in the capillaries, thus there wouldn't be passage of substances from the blood inside the capillaries to Bowman's capsule. So first of all, high glomerular pressure, and number two, the presence of fenestrations in the capillaries. Of course, you can say that the loop of capillaries is coiled and has a large surface area, which increases the rate of filtration. But we also have reabsorption. So I might ask you about significance of reabsorption, and we mentioned it before and said that how is reabsorption important? When I ask about the significance, then I am asking about the reabsorption, and when I'm asking about the importance, significance, I'm asking about the importance, and the importance of reabsorption is similar to the definition, meaning that it prevents you from losing useful substances like water and salts by returning them to the blood. What are the characteristics that of the substances that are filtered? Of course, this is another important substance. The particles that are filtered shouldn't be large. They should be smaller than the size of the pores, and they should be non-charged. Okay, so the bigger they are and the more charged they are, the more difficult their passage through filtration is. And here come two important questions. The first one is related to the content of urine. When we say that amino acids and glucose do not appear in urine, although they are filtered, but I might not say although they are filtered. You know that they are freely filtered. You know that amino acids and glucose are freely filtered in the glomerulus. However, they are not normally present in urine. If they are present in urine, then this is something abnormal. Why aren't they normally present in urine? Because they are completely reabsorbed. So because they are completely reabsorbed or 100% reabsorbed and returned to the blood. Where are they 100% reabsorbed? They are 100% reabsorbed in the proximal convoluted tubule. How are they reabsorbed in the proximal convoluted tubules? They are reabsorbed by active transport. Although they in other areas of the human body they might be passed across the membrane by facilitated diffusion using carriers, yet in the PCT they are actively pumped or actively transported from the lumen to the blood against concentration gradient using energy. Because I might ask you about active transport. So the first thing you say is that active transport is against concentration gradient, and therefore it requires energy, and that energy is derived from the ATP molecule and obtained by hydrolysis of ATP. Okay, these are just a few examples of questions that are related to that slide. But we might also have another question related to the content of urine, which is, are BCs, red blood corpuscles, and proteins? Notice that the previous question was about glucose and amino acids, but this question is about RBCs and proteins. And again, RBCs and proteins cannot be seen in urine, but for a different reason. RBCs and proteins do not show in urine because they are too big to be filtered. Not only are they too big to be filtered, but they're also partly charged, and therefore they find difficulty passing. And that's a good thing, because we do not want to lose our RBCs in urine, neither do we want to lose our proteins in urine. So the formation of urine is the result of three processes, and these three processes are filtration, reabsorption, and secretion. Which of these three processes adds substances to urine? Filtration adds substances to urine, and secretion, whereas reabsorption removes substances from the filtrate and returns them to the blood. Now, what is secretion? Secretion is essentially the opposite of reabsorption, meaning that secretion means the transport of substances. If this is the blood vessel, if this is blood, then secretion would be transporting substances from the blood into the renal tubules. Why are we doing this? Because these substances, why are we doing this? Because these substances are toxic substances. So if they are toxic substances, why haven't you filtered them? Because they were too big to be filtered or too charged to be filtered. So if you have a substance with these three characteristics, toxic, unwanted, too big, and too charged, the best way to get rid of it is secretion. Now, do we have other excretory organs in the body? Yes, we do have the lungs and the skin. What are the excretory products of the lungs? They excrete carbon dioxide and water. What is the excretory product of the skin? It is sweat, but it's not enough to say sweat. You have to say sweat. That consists of the surplus water, surplus salts, and a minimum amount of urea. What about the content of urine? Urine contains the surplus water, surplus salts, just like sweat, as well as the nitrogenous waste of protein catabolism, in addition to the metabolized drugs and hormones. I might also ask you about the coordination between the skin and the kidneys in what concerns excretion of water and salts. So you would say that in summer, because the weather is hot, there is more blood supply to the skin, there is more formation of sweat and evaporation to cool down the body, and therefore the volume of water and salts that is excreted by the kidneys diminishes and urine becomes concentrated, and the reverse occurs in winter. You might also be asked about urea, and you might be asked about the organ that synthesizes or makes urea and the organ that excretes urea. So urea is synthesized by the liver, by liver cells, and it is excreted by the kidney. Here you can also be asked another famous question, which is justify the liver converts ammonia into urea. Ammonia is converted into urea because urea is less soluble, therefore less accessible to the cells, and therefore less toxic. So you have to write the 3 points. It is less soluble in the water of plasma, therefore less accessible to the cells, or less capable of being distributed on the body cells, and therefore it is considered less toxic. So urea is less toxic than ammonia. And then this formula, this structural formula of urea indicates that every molecule of urea is made up from 2 molecules of ammonia and 1 molecule of carbon dioxide, of course, after removing some atoms, because ammonia is essentially NH3, not NH2, and carbon dioxide is essentially CO2, not CO. We talked before about the importance of the presence of urea in sweat and how urea acts as a moisturizing agent, and why do many creams contain many cosmetic creams or hydrating creams contain urea. Of course, in the anatomy of the kidneys and the urinary system, you might be asked about the famous position of the right kidney and why the right kidney is lower in position than the left kidney, because it has the liver above it. You might also be asked about the peritoneum. What is the peritoneum? The peritoneum is a membrane that lines the abdominal cavity and covers the abdominal organs. You might also be asked about the kidneys, and this is the posterior part. This is the anterior or front part of the section. This is the diagram represents a section in the human body at the region of the abdomen. This is anterior and this is posterior. So the two kidneys lie at the very rear or the most posterior part of the abdominal cavity. And there is an important question here, which is why the kidneys are considered retroperitoneal organs, because they lie behind the peritoneum, outside the peritoneal cavity. We talked before about the blood supply of the kidney, the difference between the renal artery and the renal vein. And there is an important difference that we haven't mentioned, that the renal artery is a branch of the aorta, whereas the renal vein is a tributary of the inferior vena cava. Branch, we talk about branch of an artery, but a tributary of a vein. We talk about a branch of an artery and a tributary of a vein. In such a diagram, you are supposed, sometimes you are required to label the diagram. So the outermost layer would be the capsule of the kidney. Number one is the capsule of the kidney, and then you have the outer third of the renal tissue, which is the renal cortex. And then you have the pyramids, and these are the medullary pyramids because they collectively together, all the medullary pyramids together form the medulla. And then the medullary pyramids pour their urine content into minor calyces, and then into major calyces, and then into the renal pelvis, and then into the ureter. These striations you see are the striations of the collecting ducts, and the collecting ducts pour into minor calyces. So the order is like this. If you start in the nephron at Bowman's capsule, so you have Bowman's capsule, and then you have the PCT, and then you have the loop of Henle, and then you have the DCT, and then you have the collecting duct, and then you have the minor calyx, and then the major calyx, and then the renal pelvis. So if I ever ask you, distal convoluted tubules pour their content into collecting ducts. Collecting ducts pour their content into minor calyces into major calyces, and then into the renal pelvis. You might also be asked about the hilum of the kidney, this inner concave part of the kidney, and the three structures that arise from the hilum of the kidney. And these three structures are the renal artery, the renal vein, and the ureter. So again, what is the hilum? The hilum is the inner concavity of the kidney, because it is towards the middle line. So we call it the inner concavity of the kidney. What are the three structures that are or that originate from the hilum? You have the renal artery, the renal vein, and the ureter. What is the nephron? The nephron is the functional unit of the kidney. And if you say that it is the functional unit of the kidney, then you have to explain what this means. The functional unit of the kidney means that every nephron alone can carry out the function of the whole kidney. Every nephron alone can carry out the function of the whole kidney. I might also ask you about the structures that lie in the cortex and the structures that lie in the medulla, in the nephron. So in the cortex, you have the Malpighian corpuscle, and don't forget that the Malpighian corpuscle is Bowman's capsule and the glomerulus. And then you have the PCT and the DCT. These are the structures that lie. Actually, we shouldn't say Malpighian corpuscle. We should say Bowman's capsule, because the glomerulus is not a part of the nephron. So what are the parts of the nephron that lie in the cortex? The parts of the nephron that lie in the cortex are: number one, Bowman's capsule; number two, the proximal convoluted tubule; number three, the distal convoluted tubule. Whereas the part of the nephron that mostly lies in the medulla is the loop of Henle. And concerning the loop of Henle, you might be asked about the main function of the loop of Henle. The main function of the loop of Henle is to concentrate urine. What does it mean to make something more concentrated? A more concentrated solution is a solution that has more solid particles and less liquid particles. So you might be asked about the function. You might be asked about what concentration means. You might be asked about how the loop of Henle concentrates urine. So you would say that it actively transports salt or sodium chloride out. And then secondarily to this, you would have secondary osmosis or secondary reabsorption of water. So this is how the loop of Henle concentrates urine. What helps the loop of Henle to concentrate urine is that it acts as a countercurrent system. So you might be asked about how the loop of Henle is well adapted to concentrate urine by being a countercurrent system. What does it mean to be a countercurrent system? It means that we have two arms that are parallel to, so you have two arms that are parallel to each other, that are close to each other, and the fluid in them circulates in two opposite directions. What's the significance or the importance of countercurrent systems in general? It is to maximize the function. What function will be maximized by the loop of Henle being a countercurrent system? It is the process of concentration of urine, okay? Now, what's weird about the blood supply of the nephron? What's weird is that we have the glomerulus receives from an arteriole and gives to an arteriole. Usually when we have capillaries, like the capillaries at the bottom of the slide, they receive from an arteriole and give blood to a venule. But concerning the glomerulus, it receives from an arteriole, which is the afferent one, and gives to another arteriole, which is the efferent one. Of course, since the renal cortex contains the Malpighian corpuscle, then the main function of the renal cortex, or one of the main functions of the renal cortex, is filtration. Actually, the renal cortex contains what? Contains Malpighian corpuscle, PCT, and DCT. Therefore, it can do all functions. It can do filtration in the Malpighian corpuscle. It can do reabsorption and secretion in the tubules. We have said all this, and now we're going to, and we said what concentrated is and what diluted is, and you have to know the reasons or the causes of a more concentrated urine. You can get a more concentrated urine when when you don't drink enough water, when you're starved, when you're dehydrated for a reason or another, when you sweat a lot, and you can have a very diluted urine in winter, when the weather is very cold and you don't sweat at all, or when you forcibly drink large volumes of liquid or water in a short period of time. Or another cause of diluted urine is a disease like diabetes mellitus or diabetes insipidus, okay? The filtration membranes, of course, are important. We have three filtration membranes. We have the podocyte membrane, that belongs to Bowman's capsule. We have the basement membrane in the middle, and we have the endothelium or endothelial membrane of the capillaries. The endothelium is characterized by fenestrations. The podocytes are cells possessing feet-like processes and having slits in between. So the slits are narrower than the fenestrations. These are facts you have to know. So the podocytes possess foot-like processes. The podocytes belong to Bowman's capsule. The podocytes have narrow slits in between that further control the process of filtration. The endothelium is a one layer of cells lining the capillaries, having fenestrations in between. Both the fenestrations and the slits control what passes through the membrane. They allow passage, but they control. They control according to what? According to the size and according to the charge. What is the basement membrane? First of all, where is it located? It is located in the middle between the endothelium membrane and the podocyte membrane. What's its importance? Its importance is that it is the membrane on which both the endothelial membrane and the podocyte membrane lie. But is it the only importance? No, there is something else, that the basement membrane contains collagen protein molecules that carry a negative charge, a weak negative charge, and therefore they will prevent or diminish the filtration of charged substances. The factors affecting filtration have been mentioned before. We talked about high pressure in the glomerular capillaries. We talked about the fenestrations, and here you have to mention the diameter, which is 100 nanometer, and a nanometer is 10 to the power minus 9 of a meter. Anything that is smaller than 100 nanometer will first pass through the fenestrations. Anything that is larger, like the blood cells and the proteins, will not pass. And as we said before, there are two other limitations. The first two factors, the high pressure in the glomerular capillaries and the fenestrations, are helping factors to a great extent, but the slits and the negative charge are controlling factors. We talked, of course, about reabsorption, and I might ask you about the substances that are reabsorbed in the PCT and how they are reabsorbed, or the mechanism of their reabsorption. We mentioned this before. We said that concerning reabsorption, you have glucose and amino acids. These are 100% reabsorbed by active transport, glucose and amino acids. But we also have salt and we also have water that are reabsorbed, not 100%, but only the required amount. Salts are reabsorbed here again by active transport. So glucose, amino acids, and salts are reabsorbed in the PCT by active transport, whereas only water is reabsorbed passively by osmosis, okay? And this is another diagram that recaps all what we have said. And then, of course, the transport across the cell membranes. You should know that there is passive transport and active transport. You should know the characteristics of passive transport and an example. So if you were to say the characteristics of passive transport, it does not require chemical energy. It occurs down concentration gradient. It does not necessitate a membrane. Of course, the best example is simple diffusion of oxygen, of carbon dioxide across cell membranes. But passive transport also includes, other than simple diffusion, you have facilitated diffusion. If the molecule or the particle needs a carrier or a channel, but yet the transport is passive, down concentration gradient, without energy expenditure, so it has all the characteristics of passive transport with only one difference, then it is facilitated diffusion. Why would we need a carrier? We would need a carrier for large charged molecules like glucose and amino acids. Why would we need a channel? We would need a channel for ions, whether they are positive ions like sodium and potassium or negative ions like chloride, okay? Now, notice that or note that some particles like sodium ions, sodium chloride, for example, can pass by facilitated diffusion in some parts of the human body or in some cell membranes, but in other areas, active transport can be utilized. Active transport, of course, necessitates energy, and that energy is obtained how? It is obtained by breakdown or hydrolysis of ATP. What does hydrolysis of ATP result in? It results in ADP, or adenosine diphosphate, plus phosphate, plus chemical energy that will be needed to pump the particles against concentration gradient. Of course, the third characteristic of active transport is that it necessitates the presence of a membrane. The best example for active transport in the human body is the sodium-potassium pump. First of all, the pump is a membrane protein, and it is an active transporter, meaning that it pumps particles or ions against concentration gradient from lower concentration to higher concentration. So number 3, it requires energy, chemical energy derived from ATP. How does it do this? It pumps 3 sodium out for 2 potassium in. Why? Justify the significance of what the sodium-potassium pump does. The sodium-potassium pump this way ensures that sodium is the main cation extracellularly outside the cell, and potassium the main cation intracellularly or inside the cell. And why do we want sodium to be extracellular and potassium to be intracellular? Because this ensures optimum functioning of the cells. Why is the sodium-potassium pump considered a co-transporter? Because it transports 2 ions at the same time. Why is it considered an anti-transporter? Because the direction of pumping of sodium is opposite to the direction of pumping of potassium. What is osmosis? Osmosis is a special type of diffusion. So it is passive transport. It occurs down concentration gradient from higher to lower, but here we have to say higher water concentration to lower water concentration across a partially or selectively permeable membrane. Why is it important to have a membrane? Because the membrane has pores that allow only the passage of water molecules and retain the solute. And what's the importance of this? The importance of this is to allow us to study one factor at a time. Of course, water will pass from higher water concentration or a more diluted solution to lower water concentration or a less diluted solution. And of course, it goes without saying that a less diluted solution is a more concentrated solution. We talked about the loop of Henle, and now we're going to talk about the collecting ducts. Every collecting duct receives the filtrate or the liquid from several distal convoluted tubules. The collecting ducts are the site where the final tuning of the water concentration occurs. And this final tuning, the second point you have to know about the collecting ducts is that this fine tuning of sodium concentration occurs under control of the antidiuretic hormone. What does the antidiuretic hormone do? It opens aquaporins. What are aquaporins? They are passive water channels. Why are they important for osmosis? They allow rapid osmosis. Therefore, if water passes via aquaporins in order for osmosis to occur rapidly, then this would be one type of facilitated diffusion. What happens if the pituitary gland, the posterior lobe of it, does not secrete enough antidiuretic hormone? The aquaporins would be closed. There would be no reabsorption and no returning of water to the blood, and the water will be lost in urine. Urine will be very diluted. Urine will have huge volumes, and this condition of deficiency of the antidiuretic hormone is called diabetes insipidus. It is a rare condition, but it is one important cause of polyuria. I'll stop here because you have two other videos that talk about that review actually the disorders and diabetes. So you can refer to these two videos, the one on diabetes mellitus and the one before on disorders. They are explicit and they contain many questions. And in order to avoid repetition of what I've already said, so you would be watching this video in addition to the video on the disorder and the video on diabetes mellitus. Good luck, inshallah, with your test tomorrow, and I'll be at school. If you have any question, you can ask me over the phone or relay your question to the teacher who is proctoring, and she will be contacting me. Thank you and good luck, inshallah.